We're surrounded by these incredible maps, right? We've charted the deepest oceans, the farthest galaxies. But what if I told you that the most complex, the most amazing universe is actually inside every single one of us? We're talking about the 3D world of our own biology. And that's the big question, isn't it? It's this massive challenge that's driving so much of biology today. How do we create a detailed, high-res atlas of something as ridiculously complex as a brain or a developing tumor? Well, for the longest time, our best shot at this has been, well, kind of brutal. We take a magnificent 3D organ, like this mouse brain here, and we slice it into these paper-thin 2D sections. It's like someone hands you a novel, but you only get to read one page out of every 10. You get the gist, maybe, but you're missing almost the entire story. And this really gets to the heart of what we can call the 2D problem. Scientists get these amazing, super detailed 2D images, but the slices themselves are often separated by these huge gaps. We're talking 100 micrometers apart sometimes. At the cellular level, that's like a whole continent of missing information. Okay, so you're probably thinking, why not just, stack them up in a computer? And yeah, we can do that, but it doesn't solve the problem. All you get is a stack of flat pictures with all those giant gaps still there. It's not a real continuous 3D volume, not even close. So how do we fill in those missing pages? Well, this is where things get really cool. A new computational framework published in the journal Nature Methods has stepped up to the plate. It's called SpatialZ, and it's designed to do something that sounds like science fiction. It invents virtual slices to fill in those exact gaps. Now, to get how SpatialZ works, we first got to talk about the amazing tech it's built on, spatial transcriptomics. Just think of it like a biological GPS. It doesn't just tell you which genes are turned on in a cell, it tells you exactly where that cell is. It literally creates a map of genetic activity inside a tissue. At its heart, SpatialZ works on this really elegant but powerful idea. Biology is continuous. A structure in one slice doesn't just disappear and then magically reappear in the next one. It flows, it transitions smoothly. So SpatialZ intelligently predicts what's in the gaps, kind of like how a CGI artist creates the frames between two key drawings to make an animation look totally seamless. So how does it pull this off? Well, the workflow is just brilliant. First up, it lines up the real slices we actually have. Then, and this is the magic, it uses a deep learning model to predict where new cells should be in the empty space. Next, it figures out what kind of cells they are. And finally, it generates what their gene expression probably looks like. It's literally computationally writing the rest of the book for us. Okay, that all sounds incredible on paper, right? But how do you really know if your computer-generated virtual slices are any good? You have to test it. So let's see how the researchers put this thing through its paces. And this is the mic drop moment. The researchers took a real 3D dataset, but they hid some of the slices in the middle. Then they asked SpatialZ to recreate one of the missing ones. On the left, that's the real slice, the ground truth. And on the right, that's what SpatialZ predicted. I mean, just look at that. The resemblance is absolutely stunning. And we can actually put some hard numbers on this. This chart shows the correlation with the real 3D data. Now, the original sparse slices are already really good, with a correlation of 0.98. But look what happens with the SpatialZ atlas. It pushes that accuracy even higher, to 0.99. So it's not just plugging holes, it's making the entire model even more true to life. So once they knew this thing worked, the researchers decided to go big. They took this massive dataset from the Brain Initiative, 129 sparsely sampled sections from a mouse brain. And by generating 9 virtual slices for every real one, they built a single, seamless, high-fidelity 3D mouse brain atlas with over 38 million cells. To put that in perspective, the paper says a feat like this is just not feasible experimentally. And this is where it gets really, really fun. What can you do with a perfect digital copy of a brain? It's basically like having a biological Google Earth, and it gives scientists some brand new superpowers that were pure sci-fi just a few years ago. Superpower number 1 in silico sectioning. See, when you physically slice a real organ, that's it. You're stuck with that one angle. But with a complete digital model, you can reslice it any way you want. Sideways, top down, a weird 45-degree angle, you can explore biological structures from perspectives that were literally impossible to see before. Superpower number 2 is just as cool. You can take all that data from millions of individual cells and transform it into a smooth, continuous 3D surface. Why is that a big deal? Because it lets you see how gene expression flows like a smooth gradient across an entire structure. You're not just looking at dots on a slide anymore. You're seeing the whole biological landscape in motion.
Now, obviously, this is a total game changer for brain science, but this powerful technology, it's not just for brains. So the big question is, what's next?
To prove just how versatile this tool is, the researchers threw a completely different problem at it. They went from mapping gene expression in a mouse brain to mapping protein expression in human breast cancer tissue. A totally different system, a different technology, and guess what? It worked just as well, letting them see these complex 3D patterns inside a tumor.
And this is really just the beginning. The same ideas could be used to map proteins, metabolites, or even multiple different kinds of data all at once. It opens the door to building atlases of way bigger organs, and even comparing these incredibly detailed structures across different species. The possibilities are huge.
So what's the big takeaway from all this? SpatialZ is this incredible bridge. It takes the sparse, disconnected 2D data we can get from experiments and transforms it into the rich, continuous, and fully explorable 3D atlases that scientists have been dreaming about for decades. Which leaves us with a pretty inspiring final thought. For all of human history, our own bodies have been the greatest uncharted territory. Now that we finally have the tools to map this territory in 3 glorious dimensions, what part of ourselves are we going to explore first?